Hello everyone and welcome to today's webinar. I'm Eungyung Lee, an application specialist at Curiosis. Today we are going to talk about how maintaining cell health with live cell imaging can lead to more reliable results in cell-based results. Thank you for taking the time to join us. Here's a quick overview of what we will cover today. First, a brief introduction on why stable cell conditions are so important in cell-based research. And then we will look at CellLogger, our automatic live cell imaging system, its main features and model options to match different research needs. And finally, we will walk through cellular applications, including spheroid cytotoxicity, adipogenesis, and phagocytosis. As many of you know, reproducibility has become a major concern in modern research. Even well-designed experiments often yield inconsistent results across labs. In cell-based research, one of the biggest reasons is biological and environmental viability. Even small differences in culture conditions can completely change cell behavior. So, as researchers, one of our main goals is to identify those sources of viability and control them as much as possible. That's where keeping cells healthy and conditions stable becomes fundamental. To do this, we need to manage three main environmental factors: physical, chemical, and biological. Physical factors like temperature, humidity, and CO2 must stay constant. And chemical factors include pH stability and nutrient balance, which directly affect cell metabolism. And biological factors, cell density, pesticides number, and contamination control are just as critical. So reproducibility starts with consistency in all of this, and the best way to maintain that is through real-time monitoring, which is exactly where live cell imaging comes in. Live cell imaging is a technique that uses specialized microscope and labeling methods to observe and study biological processes in real time within living cells or organisms. Unlike traditional fixed cell imaging, which only gives you static snapshots, live cell imaging, like you see in this video, lets scientists watch cellular behaviors like cell division, migration, signaling, and interaction with other cells or molecules as they happen. This approach has become essential in a lot of fields like drug discovery or cancer research. Because of this advantage, live cell imaging is now being used more and more as supporting data in scientific publications. For example, in this study on oral squamous cell carcinoma, researchers used time-lapse videos of different cell lines to visualize wound healing processes over 24 hours. These videos were included as supplementary materials, providing clear visual evidence to support the result presented in the paper. These trends reflect how live cell imaging strengthens data reproducibility and transparency, giving reviewers and readers a more complete picture of what actually happens inside living cells. When live cell imaging technology was first introduced, it wasn't as advanced as it is today, so researchers had to rely on manual methods. This means regularly taking cells out of the incubator, observing them under a microscope, recording the results, and then returning the cell back to the incubator. This repetitive process required a lot of labor and often led to significant variability in results between different researchers doing the same experiment. Plus, constantly removing and returning the cells disrupt the cell culture environments. and increase the risk of contamination. To solve these issues, automated live cell imaging equipment has been developed more and more in recent years. Most of the live cell imaging system features time-lapse imaging capabilities that automatically capture images, ensuring consistent results without variability between users. Many systems operate directly inside a standard CO2 incubator or include an integrated environmental chambers or controller, allowing experiments to be conducted under stable conditions, minimizing the risk of contamination. However, even with automation, one major challenge remains maintaining a truly stress-free culture environment. According to recent market reports, the biggest technical hurdle in live cell imaging is keeping cells healthy over long-term imaging. Even slight mechanical stress, temperature fluctuations, or phototoxicity can affect cell viability and alter experimental outcomes. This is especially critical for sensitive cell types or long-term assays, where maintaining stable condition is essential. So while automation improves consistency, environmental stability inside an image system remains the key challenge in the field. And this is where Cell Logger offers a clear solution. The automated live cell imaging system Cell Logger comes in four different models: Cell Logger Nano, Mini Plus, Pro, and Stack. With its compact design and efficient live cell imaging capabilities, it fits seamlessly into existing incubator and lab workflows, minimizing hands-on times and scaling from bench-scale studies to larger cultural operations. One of the key strengths of the Cellular series is that you can place it directly inside an incubator, allowing continuous monitoring without disturbing the cells. You can view and control everything remotely from an external PC or laptop, keeping culture conditions completely stable. For higher throughput, up to 16 units can be connected in one incubator, while only one LAN cable is needed outside for control and data analysis. This ensures continuous, contamination-free, and reproducible imaging. Another key feature of Cellular is its optic moving design. In most live cell imaging systems, the plate moves while the camera stays fixed. As you can see in the video here, that motion can introduce vibration and disturbance, leading to cellular stress or even detachment in sensitive cultures, and then in turn can affect cell growth. Cell Logger solves this with an optic moving type. The vessel stage remains stationary and only the camera moves below the stage. It keeps multiple positions stable and in focus, which is especially important for suspension cells. This setup supports cell viability and improves frame-to-frame consistency by minimizing positional drift and motion artifacts, yielding cleaner, more reproducible data. The cell logger comes with its own scanning and analysis software with a user-friendly, intuitive interface, making it easy to learn and use. It also has no installation limits, so you can install it in your personal computer and run analysis whenever you need. Let me introduce some of the key software functions. Start with time-lapse imaging. Time-lapse imaging is one of the most frequently used functions. You can set up the schedule imaging intervals depending on your experiment, every few minutes, every hour, or even over several days. The software automatically compiles those images into videos, allowing you to visualize cell behavior continuously in both bright field and fluorescence channels. And the preview record function lets you capture live videos in real time, which is especially useful for tracking fast responses or sudden morphological changes. This provides more dynamic and detailed data, as you can see in the video on the right. For experiments involving 3D structures like spheroids or organoids, the G-stacking function is essential. This feature captures multiple images at different focal plane along the G-axis, which are then combined using the G-projection functions to create a single, highly focused composite image. In the example shown here, we analyzed the GFP-tagged spheroid treated with ETHD1 dye to evaluate drug-induced cytotoxicity. We captured 15 images at 8 micrometer intervals along the z-axis and merged them using the maximum intensity projection method. The resulting image clearly highlights the red fluorescence of dead cells, providing precise data for cytotoxicity analysis. When you need to observe a large area in a single view, the stitching function is helpful. It allows you to combine multiple images into one seamless, high-resolution image as shown on the left. On the right, you can see an example how this feature is used to analyze transfection efficiency across the wide field. By capturing and stitching together multiple regions, researchers can efficiently observe and evaluate the overall success of specific gene transfection in cells. Next, the cell confluence analysis is one of the most widely used features. It calculates the percentage of the field covered by cells, and when you upload images from different time points, automatically track confluency over time and generate graphs for easy visualization. It can also measure the wound area or 3D cell area like spheroid or organoids, giving you clear quantitative results. Also, the cellular analysis software makes it easy to evaluate fluorescence coverage and intensity. It automatically detects and quantifies fluorescence signals across the field of view, simplifying the analysis of gene expression, toxicity, and so on. Cell fluorescence intensity analysis, which is called cell FL intensity, focused on measuring fluorescence specifically within cell regions rather than across the entire field of view. This targeted approach not only removes background noise but also addresses the limitation of the mean fluorescence intensity methods, where a lower fluorescence level in the background can underestimate overall intensity values. By isolating fluorescence within defined cell regions, the cell FL intensity method delivers more accurate and reliable experimental data. Now let me briefly summarize the cellular series and its lineup. First, Cellular Mini Plus is designed for basic live cell imaging among the cellular series. It has a fixed fluorescence filter and magnification, ensuring stable and reliable results for routine experiments. Next, the Cellular Pro is the most advanced model, offering a greater flexibility and enhanced features. It supports dual fluorescence filter, green and red, and allows users to change lenses, making it perfect for more diverse and complex applications. Next, the Celico Nano is a compact, lightweight digital microscope, about half the size of an A4 sheet of paper. Its portability makes it ideal for use in incubators and clean benches. With X, Y, and Z axis adjustment knobs, it allows for quick and accurate focusing, making it an excellent tool for precise and flexible cell imaging. Lastly, the cell stack is specifically designed for large-scale cell culture processes using multi-layer vessels. With time-lapse and multi-position imaging features, it helps determine the best timing for cell harvesting and establish standardized protocols. Its model is tailored to meet the unique needs of researchers, ensuring that you have the right tools for your specific applications. Now let's see how Cellular is used in practice. We will look at three applications, spheroid cytotoxicity, adipogenesis, and phagocytosis. It involves multi-step workflows that require regular checks, for example, confirming spheroid formation or tracking differentiation status. With live cell imaging cell logger, let us follow these changes in real time, capturing every stage without disturbing the cells. Then let's start with spheroid cytotoxicity assay. There is a broader shift in drug testing right now. The US FDA announced that new drugs can now be approved without animal testing. This is a groundbreaking decision because traditional animal testing has significant limitations. Not only does it require sacrificing a large number of animals, but it also has a very low success rate. Only over 5% of drugs tested on animals eventually receive FDA approval. That's why 3D cell models such as spheroid and organoids are gaining traction. Compared with 2D structures, they better mimic human tissue architecture and gradients, making them a more realistic and informative platform for testing. The cellular system supports this workflow end to end. You can monitor spheroid or organoid formation, verify quality, and then track drug-induced cytotoxicity over time, all inside an incubator with precise real-time measurements. Since meaningful cytotoxicity data starts with well-formed spheroids, let me first show how we generate them. There are several ways to form spheroids, but in this experiment, we use a 96 well U bottom plate. When cells are seated into these wells, they naturally aggregate and form rounded spheroids, as you can see here. The video below shows an example using two different cell types, MDA MB231 and HeLa. Each was each was seated at different seating densities, and spheroid formation was captured every 15 minutes using the Cellular Mini Plus. This allows us to observe and analyze the progression of spheroid formation over time. Once the spheroids were formed and quality checked, we move on to a cytotoxicity assay. For this assay, GFP expressing HEK293 spheroids were generated in a 96 value bottom plate. After formation, we treated with the spheroid with staurosporine, which is an anti-drug at zero point 0.1 and 1 micromole, and then added ETHD1, a red dye that levels dead cells. Using Cellular Pro, we acquired time-lapse images to track changes over time. As you can see here, red fluorescence increases after drug treatment, evidence of cell death, and the signal is stronger at the higher dose. We also observe a decrease in spheroid size with increasing concentration, further confirming the cytotoxic effect of the staurosporine. After capturing the time-lapse images, the next step is to quantify what we're seeing. We measured red fluorescence with the cellular analysis software to estimate spheroid viability over time. Let me show you briefly. First, just upload the time-lapse images captured from the cellular scan software into the analysis software. And then click the red light button at the top right, and then click the analysis button. The software automatically measures the red fluorescence intensity for each image, showing the result on the right. If you click the chart button, you can generate graphs showing how the fluorescence intensity change over time. Finally, you can click the export button to download all the data as raw Excel files for further analysis. Here you can see the quantitative result from the fluorescence analysis. Just like what we saw visually, the red fluorescence intensity increased over time, and the higher the drug concentration, the faster and stronger that increase becomes. This confirms that sporin induced time and dose-dependent cytotoxicity in the steroids. In addition to fluorescence, we can assess drug toxicity by measuring spheroid area. As toxicity rises, more cells die and the spheroid gradually shrinks. With the cellular analysis software, a spheroid area can be measured automatically over time. To do this, again, simply upload the time-lapse images captured with the cellular scan software. Then from the top menu, select spheroid cell under the Analysis tab, and the spheroid will be highlighted in yellow as shown here. After that, click the Analysis button on the right, and the software will measure the spheroid area for each image, displaying the result in the area section on the right. You can also export the raw data, which allows you to quantify the spheroid size based on the drug concentration. Combining red fluorescence intensity with spheroid area gives more complete quantitative pictures of drug-induced cytotoxicity in spheroids. Our next application is adipogenesis. Obesity and metabolic disease have become major global health issues, driving strong interest in developing therapies that target adipogenesis, the process of fat cell differentiation. Adipogenesis refers to how mesenchymal stem cells transform into mature adipocytes or fat cells. However, adipocytes are highly sensitive to their environment. Factors such as temperatures, CO2 concentrations, and pH variability can significantly influence how well differentiation occurs. Even softer differences in culture conditions or initial seeding density can lead to large variation in the degree of differentiations. That's why maintaining a stable and controlled culture environment is crucial. By using automated lifestyle imaging system like CellLogger, researchers can easily test and compare different conditions such as seeding densities or differentiation media in a single plate. This makes it possible to monitor cell morphology and differentiation progress in real time without disturbing the cells. Before starting autophotogenic differentiation, it is important to confirm that the cells have reached the right confluency. Here, 33L1 pre-adipocytes were seated at different densities and monitored using CellLogger Pro, which automatically captured images every hour. As shown below, the confluency varied depending on the initial seeding density from about 20% to nearly 100%. Since differentiation efficiency is strongly affected by confluency at induction, these steps help determine the optimal seeding density for consistent and reproducible adipogenesis. After verifying confluency, We induced adipogenesis using two differentiation media, with or without rosiglitazone, which is known as promoting lipid accumulation. To visualize adipogenesis, lipid 2 dye, which is a live cell fluorescence dye, was added to labeled lipid droplets. Using Saloga Pro, we could continuously track the formation of lipid droplets in real time without fixing the cells. Here you can clearly see the morphological differences between undifferentiated and differentiated cells. The differentiated cells contain numerous small lipid droplets throughout the cytoplasm. To quantify these changes, we use Zellweger's analysis software to measure green fluorescence coverage, which represents the lipid droplet area. Here, the graph on the right shows that differentiation progressed steadily over time and significantly enhanced in the logically treated group, confirming its strong effects on promoting adipogenesis. Overall, live cell imaging with cell logger lets you track adipogenesis continuously without disturbing the cells, quantifying it objectively, and quickly compare seeding densities and induction conditions on the same plate. That means you can optimize protocol faster, reduce variability, and generate reproducible time-resolved data instead of relying on a few endpoint snapshots. Lastly, phagocytosis is one of the most important defense mechanisms of our innate immune system. It is a process by which immune cells, particularly macrophages, recognize, engulf, and digest foreign materials such as bacteria or particles. Here you can see the typical sequence: recognitions, engulfment, phagocytomaturations, and digestions. To study this process experimentally, we used the raw 264.7 macrophage cell line, which originated from mouse monocytes. These cells can be stimulated with LPS or lipopolysaccharides to activate their immune functions and enhance phagocytic activity. Traditionally, phagocytosis assays relies on endpoint methods like flow cytometry, where only the final uptake can be measured. But with the logger, we can visualize the entire process in real time, from initial recognitions to bit uptake, providing far more dynamic information. Before testing phagocytosis, we first monitored how monocytes differentiated into macrophage under LPS stimulation. Using Cellular Mini Plus, we captured images every 15 minutes in bright field to track these morphological changes. Over time, the round, loosely attached cell began spreading and adhering more firmly, adopting a spindle-like macrophage morphology. You can see that LPS-treated cells shows a much higher confluency, about 88% compared to about 60% in the untreated group, indicating enhanced proliferation and activation. This differentiation step is essential since only mature macrophages show strong phagocytic behavior. Once the macrophages were activated, we added green fluorescence microbeads to visualize phagocytosis in real time. With Cellular Mini Plus, we could continuously observe the macrophage migrating toward the bead, engulfing them, and enclosing them inside the cytoplasm. This sequence, captured every 15 minutes, clearly shows how LPS-stimulated cells exhibit more active movement and higher bit uptake than control. Interestingly, after bit internalization, we even observed a division of engulfed bit into daughter cells during mitosis. Taken together, these observations shows that cellular stable life cell imaging allows researchers to track highly dynamic immune responses that are otherwise difficult to capture with conventional microscopy. In addition to the applications we have covered so far, Cell Logger allows you to dynamically observe various cell types across a wide range of experiments, as you can see here. If there is something I didn't mention today, please feel free to contact us. Then we'll be more than happy to discuss the different application with you. Thanks again for being with us today. If you have any question about the webinar, please feel free to drop a message through our website or email.